Okay, so before we begin working on our before we begin working at our lab stations, we want to review some of the stuff about microscopy. So first off, the size of the fields of view, which would be this distance across for the different objectives. So 4x is scanning. 4x, if you remember, is 5,000 microns. 10x is 2,000 microns, and 40x is 500 microns. So in this problem down here, it says if I wanted to move this star to the center, this direction, what direction would I move my slide? So my slide, I would have to move, so if this was my slide on my screen, I would have to move it. If I want that to move, my image to move to the right, I have to move my slide to the left. If this star is being viewed at 10x, what is the size in microns? Well, if I have that arrow that I can see on my field of view, in my eyepiece, it looks like that. Can use this to figure out roughly how many of these would fit across the screen. So looking at this, I would say about 2.5 of those stars would fit across that arrow, which means about 1, 2, 3, 4, 5 of those stars would fit across this screen. And I can kind of use my smartboard to verify that. So I'm going to do a clone, move the clone here, and continue cloning it. So 2, 3, 4, and it looks like 4.5, but you know what? I guess I can say 4. Maybe I'll stick with 5. So if I'm going to say 2,000 divided by 5, that would be 400 microns, because I know that this, it's saying that this is 10x, so that means this distance here is 2,000 microns. Or I could say 2,000 divided by 4, which is equal to 500 microns. So either of these two would be an acceptable answer. Now, the reason why I want to review that is because in this lab, what you're going to do is you're going to be looking at examples of fungi, protists, plants, and animals. And for each group, you're going to sketch the specimen on a prepared slide or a wet mount you must prepare. You're going to be provided textbooks, but really what I'm going to tell you to do is use your iPads and Google to identify structures, answer the analysis questions at the conclusion. Each of these will be worth 3 points. So you want to draw a single cell. The size of the cell is indicated, should be indicated in microns. That's 1 point. The parts are labeled 1 point. And then this is talking about your drawing. Your drawing should look like the cell. It doesn't have to be perfect, but it should look like the cell. It shouldn't just look like a bunch of scribbly lines. None of the cells will look like a bunch of scribbly lines. So the first, as it says here, this will be done in class. So I'm showing you these. These are the 3 different types of prokaryote cells. So you have a rod shape, sphere shape, spiral shape. You can see this is 2, 1, and 4 microns. So they're incredibly, incredibly small. And these are the different parts that you can see. If you are drawing from this, you'll want to pause this so that you can draw it. This is a fungus, and we're going to make a slide together of the yeast in class. Fungus, as it says, can be one-celled like yeast or multicellular like mold. So this is a slide of what mold would look like. Mushrooms underground, the underground portion of mushrooms would look like this. All have cell walls, and many of their cells are long and skinny. So when we look at yeast under the microscope, it's going to look like this. And they're incredibly small, and we're going to estimate that somewhere in the neighborhood of 100 of them could fit across the screen. Now, what you see here is a cross section. If I cut this open, the wall kind of blocks everything, but if I cut it open, I could see it had a nucleus and all this other stuff inside. And if 500, if I'm looking at this under 100x, or sorry, 40x objective, I know that this distance across the screen is going to be roughly 500 microns, and I'm saying I can see 100 of these. 500 divided by 100 is going to equal 5 microns, and that's what we are estimating the size of the diameter, the distance between here and here. You will need to do the same for the other ones. So again, yeast is incredibly small, similar in size to some of the bacteria. For the protists, as it says here, these are not really a group. The only thing that they have in common is that they are all eukaryotes, but some are more closely related to plants, animals, and fungi than each other. So again, you're going to want to use either the textbook or the pictures to draw this. As it says, the euglena on the slides that I give you are pink stained cells. Amoeba are pink or purple stained blobs. And again, if you Google the images that you'll see, so it'll look kind of like this. You'll want to add this picture of the flagella, and you're going to identify the length, and then these are the different parts that you're going to also add. Okay? Well, I want you to see if you can figure out these answers. And for plants are all multicellular. And there's 3 things that they all have in common, which we've already gone over. They have cell walls, they have chloroplasts, and they have the central vacuole. You definitely need to know these 3 unique features. For the plants, we're going to be looking at 3 different ones. The leaf mesophyll is the ligustrum leaf cross-section. I'll show you what that slide looks like in a little bit. The chloroplasts are going to be stained pink. The onion root, the nucleus will be stained dark blue. Each of these is a single cell, so this is basically what the onion will look like. This is what the ligustrum looks like, and if you zoom in on it, this is what you should see. If you zoom in on one of those, that would be like a mesophyll cell. Finally, the guard cell you have seen before in the in the microscopy lab. You're drawing the 2 guard cells. Those are the 2 things that surround the stoma, the pore. And then you want to answer this question over here. Finally, for animals, these are part of multicellular organisms. They have no cell walls. For the nerve cell, you're looking at the multipolar nerve or spinal cord. What you're going to be looking for is something that looks like that, and the different parts to label. Again, you can either use this textbook or use Google. A cheek cell will look something like that, with these different parts that you have to label. Oops. Nerve cells pass signal chemicals to allow bodies to be coordinated. Epidermal cells are another type, connective, and then muscle cells are ones that shorten. So the ones you're going to be drawing will be a nerve cell and an epidermal cell. Cheek cell's epidermal. These are the slides that you're going to be provided with. And then for the cheek cell, you'll need to make a slide. This is the steps of making the slide. So you add a drop of water to the slide, use the toothpick and rub the inside of your cheek gently, rub the toothpick in the water, add a small drop of dye, then apply the cover slip. Again, what you'll see, the cheek cells when you first see them will be really, really, really tiny. They'll look like little blue specks, kind of like that. And then you need to zoom in on them and zoom in on more. And then by the time you get all the way up to the highest power, they'll kind of look like that underneath the microscope. But again, you want to draw them so they look like that big. So again, that's about how big they'll look when you see them underneath the microscope. And that's kind of what you'll see. That's an actual picture of a leaf or of a cheek cell. Okay. Remember, you can rewind or pause at any point so you can review what you need to put in this lab.